ครับวันนี้มองโลกมองเกษตรนะครับจะพาท่านผู้ชมมาอยู่ในงานเขาเรียกว่า Global Cassava Sustainability Forum 2025 นะครับก็จัดโดยสวทชนะครับ Biotech แล้วก็ร่วมกับทางหลายๆ องค์กรนะครับแต่สิ่งที่อยากจะให้โฟกัสก็คือมี2 Professor นะครับที่จะมาพูดในงานนี้นะครับท่านหนึ่งก็เป็น Professor วิลแฮมนะครับจากชุนชิง University เนี่ยจะมาพูดถึงเรื่องเกี่ยวกับการใช้ไบโอเทคโนโลยีนะครับในการเขาเรียกว่าปรับปรุงพันธุ์ให้แม่นยำทำอย่างไรแล้วก็เป็นผู้ค้นพบมาร์เกอร์ CMD2 นะครับเดี๋ยวมาดูซิว่าเขาคุยกันเรื่องอะไรนะครับ
Our next speaker will address one of the most critical aspects of cassava sustainability. Dr. Willie Grusum is currently professor and Yushin Fellow at National Chongqing University in Taiwan since 2017. Dr. Grusum is an elected fellow of the American Association for the Advancement of Science and the American Society of Plant Biologists, and he has served as president of EPSO and the Global Plant Council. So please welcome Professor Grusum, and he will speak on innovation in cassava cultivation, molecular precision breeding of cassava for novel traits, disease resistance, and yield increase. Thank you.
Good morning, ladies and gentlemen. Thank you very much for the organizers for inviting me to the Global Cassava Sustainability Forum 2025. It's really a great honor to be back in Thailand for this meeting. I want to spend the time this morning to give you a few examples of what I think we have in our genetic and molecular toolbox to increase the quality, the resilience, and also the productivity of cassava. So I've been working with cassava now for about 25 years. So what you know is cassava is of course very important, not only in Thailand but across the globe. I mean there are almost 1 billion people living and eating cassava. I've been working in Taiwan now since about 8 years in Taiwan. Taiwan used to produce a lot of cassava, but now they only use cassava starch for bubble tea, and that's about it. And but you know that cassava faces still quite a few challenges. Breeding is difficult, it's time-consuming. The cassava genome is highly heterozygous, which makes breeding challenging. The yield in farmers' field is often low. That's not necessarily the case here in Thailand, where breeders and farmers are very successful growing cassava with high input. So there is still an economic yield gap that we have to close. Weeding is time-consuming. You all know that. Here in Thailand, it's not an issue. I mean, you know how to deal with this, but if you go, for example, to sub-Saharan Africa, that's a different story there. Clean planting material is often not available, so that makes it also challenging. There's no seed industry. I mean, the large agricultural seed companies, they're not really interested in cassava. Susceptibility to viruses and bacteria is also, bacterial diseases is also an issue. And finally, the nutritional value of cassava has a lot of starch, but if you think about many people eat cassava as a staple food. They eat cassava in the morning for lunch and in the evening. So they get a lot of calories, but not necessarily the micronutrients that your body needs. So in the last 25 years, we have been developing various technologies for improving cassava. We've, for example, used precision genome editing combined with speed breeding, and I will give you an example of this, to develop new cassava quality traits. We have increased micronutrient content in storage roots. Resistance to cassava virus diseases is really a challenge. I will give you one example of a very powerful disease resistance gene that breeders and farmers can use. Increasing nitrogen use efficiency and oil content of storage root is something we are very interested in. I can tell you I won't have time to talk about it, but we have increased oil content in cassava root, not to the extent that you can use it as for frying cassava directly in its own oil. That doesn't work yet, but we can increase oil content. We are trying to increase cassava starch content and production, and we have been in the last few years also very active in producing high-quality cassava genomes that can be used for breeding. So in the next few minutes, I will give you 3 examples of the research we are doing to advance cassava productivity and quality. So the first example is how can we increase the starch quality? So for example, having a waxy cassava starch, so cassava starch free of amylose, that's of high interest to the industry and also for cooking. I mean, of course, you can do that by conventional mutagenesis, as for example, Hernan Ceballos has done. He has produced waxy cassava in Colombia. We decided to do this by precise genome editing. So you know that how amylose is formed. So there is an enzyme called the granule-bound starch synthase. There's another protein that targets this enzyme to the growing starch granule to produce the amylose. So we targeted both of these proteins, or the genes for these proteins, to knock them out because that should give us amylose-free cassava starch. So we have combined the CRISPR-Cas technology with another strategy to make cassava flower early so that we can actually do what we call the speed breeding, so breed cassava more quickly. And for that, we use the Arabidopsis flowering locus T gene that allows us to produce early flowering cassava. So in this case, we have actually managed to knock out the cassava genes. So, okay, here we go. And you see that these are several knockouts that we have produced using the CRISPR-Cas technology, and that has given us cassava lines that are now free of amylose. So you see this here, the starch, when you stain it with iodine, it doesn't stain blue anymore, which means it has no amylose. And you can also see this, I cannot work properly with this. You can see this also, these lines do not produce the granule-bound starch synthase. So the technology really works. It works quite fast because now in the greenhouse we have flowering cassava. We can make the genetic process to remove the CRISPR-Cas enzyme that has given us the mutation. And we made the mutations actually also homozygous, so that in the end, we have a time savings gain of about at least 3 years over if we would have produced the waxy cassava with a conventional breeding strategy. That is, for example, mutation, chemical or radiation mutation, and then selecting for the mutants. So I think from my perspective, the new breeding technologies, including the genome editing technologies, really provide also for cassava, especially for cassava because it's so highly heterozygous, a great advantage. And I encourage the Thailand government also to look into this. I know that there are now programs funded in Thailand in genome editing, but I think cassava has a great potential for this technology. So cassava mosaic disease resistance is also a big problem because we are losing a lot of cassava yield, especially in Africa, to the cassava viruses. And cassava mosaic disease is now spreading in Southeast Asia, so that's a big problem. We do not have a lot of resistance to these viruses in cassava. So there is this what we call CMD1, that's a polygenic recessive resistance in cassava, but for breeders it's very difficult to use. So what has been powerful is this CMD2 resistance, which is monogenetic dominant, and this resistance was first actually found by farmers in the field. So they identified cassava plants that all of a sudden were resistant to the virus, and we were doing a lot of work with cassava transformation and so on, and at some point we transformed cassava plants that were resistant to cassava mosaic disease, and we lost our resistance. And then the Gates Foundation came and said, guys, you cannot do this. This is not good. So find out what this CMD2 resistance is. And this was work that we did in my laboratory, where we actually did a whole genome variant analysis over many, many different cassava cultivars, resistant and susceptible cultivars, to look what is actually the gene that gives this resistance. And the only gene that segregated with CMD2 resistant was DNA polymerase delta 1. So that's a DNA polymerase that is necessary, of course, for replicating the cassava genome, but the virus also uses this DNA polymerase to replicate its own genome. So, and what we identified is that the DNA polymerase in cassava had a mutation, a single mutation, that caused these plants to be resistant to the virus. Okay? So that makes it a very, very powerful resistant gene, because we know exactly where the resistance is. It's in the domain of the DNA polymerase delta that is active in the DNA replication. And I don't have time to show you all the data, but I can tell you that this enzyme introduces mutations into the Gemini virus during DNA replication. And that's something that the virus cannot overcome. Okay? So if the virus is mutated, that's it. It's the end of the virus. So it's a very, very powerful resistant allele that we should use, that we should deploy in cassava. Now, the only problem, of course, is that the gene has to be heterozygous. It cannot be homozygous, because then it's also lethal to the host. Okay? We have identified these mutations in DNA polymerase delta now in many other crops that correlate with resistance to these begomoviruses. So it's a very, very powerful resistance. We patent that. So these are patents that are filed in the US and internationally. And if anybody is interested, these patents can be licensed. So we patent that not to make money. We want to make sure that this technology is available. And here, again, genome editing is a great tool to introduce these mutations. And I can tell you exactly which mutations to introduce to make the cassava resistant to begomoviruses. And if you then, of course, propagate the cassava vegetatively, you have no problem in maintaining that resistance. It's stable in the field. So the last example that I wanted to show you is spending just a few minutes to talk about our cassava source sink project, in which we are trying to increase cassava starch and storage root yield. And this is an international collaboration between many different labs. One of our collaborators, Uwe Rusche, is here. I mean, he will mention this project later. I don't see him on this slide right now, but if he stands up and shows you his face, then you will probably recognize him on the screen or somewhere. Or Uwe, you can point out where you are. But anyway, so this project, in this project, we are trying to increase cassava yield, and it's actually an engineering project. It's very interesting because it relies on a lot of information that we have from many different cassava cultivars that differ in their starch and sucrose content, and we are trying to understand which genes and which factors are necessary to drive the cassava starch production and storage root yield. Because storage root yield is not only starch production, but it's also a combination, of course, of storage root growth and the overall productivity of the plant. So in this project, our idea is to facilitate photosynthesis, so increase the activity of the source, the leaf, then facilitate the transport of the fixed carbons, so the sugars, and eventually use the sugars to convert them into starch and bulk up the productivity of the root. And that's a cycle that involves, of course, a lot of information, a lot of knowledge, but also testing the cassava plants in the field. And we have developed a very exciting strategy that we essentially take the genes that we are interested in, put them into cassava, and immediately go to the field and analyze the plants in the field. So there's no greenhouse involved. We look at the new traits in the field. So that, of course, this kind of engineering technology requires an extensive toolbox. And so we have been working also on creating this toolbox that's available to all of you, to all the cassava breeders, but also the molecular biology technology community. It's also patented, but these promoters and the tools that we have developed and are developing for cassava are readily available to anybody who wants to use them. So in this project, we are doing a lot of field testing. So we have large fields. For example, this year, we are growing over 6,000 cassava plants in Taiwan. Why in Taiwan? Well, Taiwan is actually very good. I mean, it has a perfect climate for cassava, and we don't have virus diseases in Taiwan for cassava. So that makes it the destination to test your cassava plants. And so we use a lot of technologies. Also, sorry, that was too fast. Let me go back. A lot of technologies in the field, either remote sensing or remote imaging of the cassava plants to actually look at their growth and figure out early on already how storage root bulking, how the growth might affect storage root bulking. So it's very interesting technology that our partners at the Forschungszentrum in Jülich in Germany are developing. And then, of course, we do a lot of photosynthesis measurements in the field also to look for those lines that have an increased photosynthetic capacity. So in the end, what we are looking for is in this circle, okay? So if this is your mean cassava growth, we are looking for whatever has been improved, where we have higher yield in the field, and then we move forward for that. So eventually, of course, I mean, we hope that that these new varieties or these new lines end up in the farmer's hands for increased productivity. I will just give you one example very quickly that this technology actually works. So in this particular case, we have used a calcium channel protein to increase cassava yield. We know that cassava uses a lot of potassium when it grows. So by introducing this non-rectifying or potassium channel, I'll keep it simple, potassium channel, it's a special potassium channel that was mutated. When we express this in cassava, what we find, or the idea is that it should accelerate the flow of sucrose in the cassava. Okay, so because you have that potassium channel that generates power to produce, to accelerate the flow of sugar in the stem, and using positron emission tomography, so PET technology, we can actually show that the velocity of the sucrose flow in cassava is increased. So that's also technology that's been developed at the Forschungszentrum in Jülich that they're using. And by doing this, we see that we have now cassava varieties that have an up to 30 to 40% increase in yield. Okay, so that's great. It's patented, but of course, technology, genes, everything is available for anybody who wants to use it. Okay, so I hope with these few examples, I've given you a little bit of insight into what is in our molecular and genetic toolbox that we can use to improve cassava. Of course, breeding is also still going to be a key factor in improving cassava yield and productivity. Not so much quality, because I think that's really where the new breeding technologies are going to have a big role to play in improving cassava yield.
Thank you very much, Professor Grusum. The floor is now open for questions. If not now, at the moment, I have one question for you, Professor Grusum. From your very detailed and technical content that you just presented, has the molecular biology kept up with the spread of the, for example, cassava mosaic disease right now, for example, in Southeast Asia?
Yes, I mean, of course, we have technology available now. I mean, we have varieties already that are resistant, and they can be used in breeding. Okay, the breeders know how to do that. But I mean, if you don't want to use breeding, if you don't want to spend time, if you don't want to introduce foreign germplasm, for example, here comes CRISPR-Cas. That's a technology that we can deploy immediately to develop that kind of resistance. Okay, and that's being done now. I mean, we are using CRISPR-Cas. We are using new breeding technologies, and in many other areas. We do this in rice now. So I really encourage you to look into these technologies and make sure that the breeders have them available, and help the breeders to introduce these mutations. It's really not going to be that difficult.
Thank you. Any more questions? Oh, we have one hand raised over there.
So, I mean, you've made some real advances using genetic engineering and gene editing, and that's great, but I was wondering what regulatory challenges you face trying to get some of these plants out to consumers.
So that's a very interesting question. So, for example, the waxy cassava, I mean, the amylose-free cassava, there's no trenching in there, right? So, I mean, if we look at most countries that have now deregulated the so-called site-directed SDN1, site-directed nuclease 1 events, I mean, for them, no problem. In EU, hopefully it's not going to be a problem, because the new EU regulation, under the new EU regulation, that would also be a variety that should be available. Thailand, I know it's still under discussion, but I hope that the Thailand government will also look into the new breeding technologies. Because you see, I mean, you have mutations, there's no trenching there. I mean, you know how the mutations were introduced, but if there's no trenching there, it's not GMO, okay? I mean, these mutations can also rise spontaneously, they can rise by conventional mutagenesis, all of that is not regulated, okay? So hopefully And that's my plea to the Thailand government. Hopefully, the Thailand government is enlightened to say, yeah, I mean, these new breeding technologies have a great potential, not only for cassava, but for many other crops.
Thank you. Any more question? At the back.
Thank you. That was very interesting. I'm coming from the other side. I normally have my boots in mud rather than looking at gene technology. But I was wondering, once your research has found basically the right answer, the right species, the right genetically modified species to plant, would it be possible to reproduce that through natural selective breeding means, and that way you could circumvent the restrictions?
Yeah, I mean, that's also a very good question. I mean, of course, there's been a lot of effort that's also supported by the Bill and Melinda Gates Foundation to look into cassava germplasm and see what you can do. I mean, how you can reproduce like a 20, 30% or 40% gain in yield by natural variation. And I mean, the next gen cassava program, for example, I mean, Hanan, I don't know if you're part of the next gen cassava program, but I mean, the breeders in that program can tell you that's been very disappointing so far. Okay, so the genetic gain that has been seen is somewhere around 3 to 5%, which is really not that significant that you can engage in a really powerful, I mean, like the maize breeders have done over the last 100 years. It's going to be very difficult in cassava. So from my perspective, I think we have to use a combination of, I mean, breeding is still going to be very important. There's no question about that. Okay, but we have to use also that in combination with new breeding technologies to introduce new traits into cassava.
Thank you very much.
ถ้าหากท่านได้ฟังของดรวิลแฮมนี่ก็คือน่าสนใจตรงที่ว่าเขาเป็นนักไบโอเทคโนโลยีนะครับก็คือคนที่ทำทางด้านพวก GMO หรือ Gene Editing นะครับเรียกว่าการปรับปรุงพันธุ์โดยอย่างแม่นยำนะครับดูซิว่าจีนตัวไหนนะครับมีคุณสมบัติอย่างเช่นคุณสมบัติในการต้านทานโรคใบด่างมันสำปะหลังทำอย่างไรนะครับเราให้แม่นยำขึ้นสามารถแก้ไขจีนในตำแหน่งไหนเนี่ยก็จะทำให้การปรับปรุงพันธุ์ได้แม่นยำและเร็วขึ้นนะครับก็หวังว่าจะเป็นประโยชน์ต่อท่านผู้ชมครับ